In just a minute or so. Good afternoon, everybody. Welcome to this afternoon's webinar. My name is Trevor Perrier, and I'll be your host today. I'm a bit new to this, so forgive me if it doesn't go quite as smoothly as you might be used to. Going well so far. I can't manage to move the slides, but there we go. I'm sorry, I can't manage to progress the slides. No. Is that worked?
Yep.
Okay, thank you. Apologise for this. As I said, there may be one or two technical hitches this afternoon. Let me start by just mentioning one or two housekeeping items. The talk is going to be recorded, and if you want to refer back to it later, you can see it on the CATS website. Please mute yourself during the talk so that we can hear the speakers clearly and there won't be too much background noise. There will be a Q&A session after the talk. So if you'd like to pose a question to the speakers, please use the chat function to write your questions down. And I will read out the questions, or as many as time permits, at the end of the webinar. And lastly, after the webinar, we will send you an email asking you to fill in a very short survey. This is extremely helpful to improve the programme. So please do fill it in and send it back to us. So just a few words about the Cambridge Academy of Therapeutic Sciences. This webinar is part of the Gateway to Translation series of events, which is organised by the Academy of Therapeutic Sciences. And CATS has been established to bring together scientists across the university who are interested in the discovery and development of new therapeutics. Its aim is to stimulate wider engagement across the university in this area of research, to unlock the potential for new therapeutics discoveries that might arise from research undertaken within the university, and to help university scientists develop partnerships with industry and other external bodies to the benefit of both parties. Part of the CATS initiative is the Industry Experts in Residence programme. And this gives you access to independent experts who can help advise you on translational research projects such as drug discovery, diagnostics, devices, and other advanced therapies. The advice that the experts can give can be both scientific and commercial, and many of them are experienced in raising funding from a variety of sources. I'm part of this programme, and I joined the team just before Christmas. My expertise is in small molecule drug discovery and development, which is particularly, but by no means limited to medicinal chemistry. And you can find out more about my expertise and experience, and indeed that of all the other experts on the programme, by looking at the CATS website, and this is the URL of the CATS website here. And the Experts in Residence programme is on the front page. You simply click on the tab, and that will take you through to find out more about the experts that are available to you. For today's seminar is going to be given by a team from DomainX, and it's my pleasure to introduce them to you. First of all, Kat Stace. Kat is a biochemist and cell biologist who obtained her PhD from the University of Cambridge. Kat then spent 10 years working in early stage companies in the Cambridge area, developing peptide drug discovery platforms in companies such as Isagenica and Bicycle Therapeutics. And Kat joined DomainX late in 2019. Alec is an acid biology scientist with over 4 years' experience of small molecule drug discovery research. He obtained his PhD in London and subsequently joined DomainX. He's an expert in SPR and more recently the GCI technology, which has been adopted in the Cray-Optimus Wave instrument that I dare say we'll hear more about later today. Alex conducts fragment screens and kinetic affinity determinations using these technologies. And finally, Jana obtained her PhD in molecular and structural biology at the University of Cambridge, and she joined DomainX in 2014. And she's worked on a wide variety of targets, including kinases, epigenetic targets, and protein-protein interactions. And Jana's been responsible for establishing the microscale thermophoresis platform at DomainX and was heavily involved in developing that company's fragment builder technology platform. So with that introduction, I'll hand over to Kat, who I think is going to kick off the presentation on behalf of DomainX.
Thanks, Trevor. Yes, I'm just going to share my screen here. Let's see. Okay, can everybody see this? Great. So thanks, Trevor, for the introduction. So we will discuss some of the biophysical screening methods that are available to us at DomainX and elsewhere in this talk today. First of all, I wanted to just set the scene with a few words about who we are at DomainX and where the biophysical platforms fit in with the drug discovery process and what we do. So DomainX is based at Chesterford Research Park, just outside of Cambridge, and we're about 20 years old now nearly, and have worked predominantly in the small molecule space, although with our growing bioscience capabilities, we now work as much in biologics as anywhere else. We have a focus on innovation and creativity, and that's why, you know, we do look to work with these cutting edge technologies in biophysics and other areas. And so we have a strong publication record with both patents and peer-reviewed articles. We have all of our staff at one site, and that really helps with how we work as one big team, and you know, the shared mindset with a lot of groups in academia that we work with, and also in early stage biotech as well. So we're about 70 scientists on site, and we have chemistry and biology under one roof. That's loosely divided into our protein science team, our assay biology team, who you're here from today, and our medicinal chemistry team. So what do we house together? And this is all part of the early stage drug discovery process. We have chemistry, so computational chemists, medicinal and synthetic chemists, and analytical chemists. In assay biology, you can see a photo of Jana there as well, biochemistry, biophysics, and also cell biology, so a lot of custom cell assays, for instance. And our protein science team that generate a lot of proteins for, say, you know, target materials going into the drug discovery process, and X-ray crystallography to support these programs with the structural insight, and also a couple of other novel technologies as well for difficult targets. So it's all part of this bigger picture of the discovery process itself. And there on the right-hand side, you can see going from the gene of interest to producing the protein construct, so your target material, and then actually generating chemical hits, and then moving that forward through medicinal chemistry to land upon your preclinical candidate. So this summarizes what we have in-house at DomainX and really contextualizes where the biophysical platforms fit into this process overall. So, you know, biophysics is important, you know, right from the get-go, really. So you're producing your protein material, your target material, it's important to characterize that properly, and that can be done with a lot of biophysical techniques. And then thinking ahead to actually generating novel hit material, for instance, you know, via virtual screening or fragment screening, biophysics is key, whether that be, you know, looking at your compounds that come out of your virtual screen, by an appropriate biophysical technique, or actually using a biophysical technique to screen a fragment library. For instance, we have our own fragment library at DomainX. And then, of course, orthogonal screening to validate those hits, and then actually, you know, driving the downstream hit to lead and lead optimization campaign. But of course, there are other aspects as well. As I say, you know, we work with biologics too. This describes the discovery pipeline. But one of the things we want to be is flexible. We focus on customization and collaboration. So, you know, biophysics is applicable to other aspects of drug discovery as well. So who do we have? And obviously you'll meet a couple of members of our assay biology team today. We have about 70 staff and a concentration on bringing in expertise and quality science And hence, that's why we focus on these sorts of techniques and developed a very strong biophysical suite. And bringing in industrial experience and a breadth of expertise as well. It's one thing to have the scientific capability and the equipment, but also, we want to be drawing from different experiences, different perspectives, insights, and also the know-how that, around different target classes and therapeutic areas that help join all this together into a seamless program. So we embrace creativity and innovation at DomainX, and the biophysical capabilities is just one aspect of that. So just to give you a flavour here for the other things that can drive this innovation, as well as the cutting edge technologies, we have things like novel products, new services that actually complement the biophysics and our other capabilities such as crystallography, providing unique offerings, for instance, combinatorial domain hunting to actually generate material for otherwise hard to express proteins, because of course without these materials, we need, you'd be struggling to set up a screening platform. And also creating bespoke solutions. So in biophysics, biochemistry, and elsewhere, actually looking about what is the right assay, what is the right platform, what is the right readout to actually move that project forwards effectively and in the right direction. And of course, we work with trusted alliances as well. We recognise that we can't do everything all ourselves, so we have some trusted partners. And we focus on addressing things that are unusual, unlocking challenges, thinking objectively, bringing together that fresh perspective, and working collaboratively with groups, academia and industry alike, to create effective solutions and generating quality science and integrity in a results-driven fashion, and maximising chances of success for these programs. So what do we have in terms of our biophysical suite at DomainX? And we're very privileged to have such a wide range of platforms at our disposal. And many of these are real, cutting edge technologies, state-of-the-art equipment. So we'll talk through these today in more depth so you can understand the science behind them, the technology, and also where the applications fit in. But we have a couple here, for instance, the nanotemper microscale thermophoresis platforms, where we're an early adopter of that. And also more recently, investing in an SPR equivalent called GCI through acquisition of the Creoptics Wave equipment. And there are some other techniques as well that we have to support this, creating a broad suite. So let's hear a little bit more about those. And I shall pass over to Jana now to go through some of these techniques in more depth.
Okay.
Do you have control of the screen, Jana?
Yes. Okay. So we wanted to start with by introducing why we like to use biophysical techniques in drug discovery. So they're very useful in measuring direct binding of compounds to a target protein. So they give you an information about the target engagement. And that can be very helpful to aid interpreting data from more complex assay systems, such as cellular assays or more complicated biochemical assays. And we also like them for targets where function might be unknown or the substrate might be unknown, or for compounds or antibodies that do not directly impact the function of our target protein. For example, fragments, which quite often have a very weak affinity and might not yet impact the biochemical activity of your target protein. They quite often are also independent of artifacts that you might see with other more complex systems, and they're very highly sensitive methods. And as I said, they can measure very weak interactions that we do see with fragments. And what we get are affinity data and on-off rates, and these can be extremely critical for other activities in the drug discovery process. For instance, when we want to produce crystal structures, or when the medicinal chemists want to improve the features of the molecule that we're working on. And so at DomainX, we use biophysical techniques quite heavily in our hit ID stages, and there we've got two platforms that Kat mentioned: virtual screening and fragment screening. Sorry. There we go. So we'll do a virtual screen. That's a computational method where we use over 2 million compounds and computationally see whether they can bind to our target of interest. And then we'll get a hit list of usually around 1,000 to 2,000 compounds, and only those then will be screened in a biophysical wet screen. And this has the advantage of significantly reducing the costs compared to a high throughput screen. And we'll usually only screen compounds that you can actually purchase. And then we'll take them through, any hits we'll take through an orthogonal binding assay before we will put them into a biochemical or cellular assay. We also use biophysical techniques for fragment screening. Historically, hit ID was quite often done using high throughput screening, and compounds here would usually have a molecular weight of over 400. And this means that you have to screen over 200,000 molecules to cover sufficient chemical space, which is very time consuming and expensive. Fragments usually are significantly smaller, only about 150 to 300 molecular weight. And that means to cover similar chemical space, we'd only need to screen about 1,000 compounds, again, making it much less expensive and faster to run. Additional benefit is that fragments can give you a better chemical starting point. However, fragments are very weak binders, and therefore we have to use biophysical methods to actually measure and identify hits. Once we've run a primary screen, and this can be done by SPR, MST, or DSF, we'll confirm them by orthogonal methods. And if possible, we'll look at crystallography to obtain, to confirm that these fragments bind in the place that we want them to bind, before we improve them and take them into biochemical or cellular assays. Let's talk about some of these biophysical techniques that are out there, and we'll talk about the most popular ones. So microscale thermophoresis, differential scanning fluorometry, nuclear magnetic resonance, surface plasma resonance, and isothermal titration calorimetry. However, these are not all techniques that are available, but we'll focus on these today in the interest of time. So to start with, we want to explain microscale thermophoresis. So this is a relatively new technique that's been around for a bit over 10 years, and it measures the strength of the interaction between two molecules by detecting the variation in fluorescence signal as a result of an infrared laser-induced temperature change. So the variation in the so one can correlate the binding of the ligand to the fluorescent target with the range in the variation in the fluorescent signal. And how do we do that? We measure the fluorescence of the labeled protein in the at assay temperature, and the fluorescent signal of a protein that is ligand bound or unbound will be the same. But once we start the infrared laser and the temperature change is induced, the fluorescent signal between the bound and the unbound state will be different. And we can use a titration series of our ligand and then take the difference in the fluorescent signal between the before and after temperature change points and plot these against the concentration and thereby derive our dissociation constant. But what does actually contribute to the difference in the fluorescence change that we measure? So there's two effects. The first one is called thermophoresis, and that's a directed movement of the molecules within a temperature gradient. and that changes the concentration of the fluorescent molecule at the point that we are observing. And the extent of that concentration change is dependent on, depending on the overall size of the molecule and charge and conformation shell. And the other factor that contributes to this fluorescence change is called temperature-related intensity change, or TRIC for short. TRIC has three principles that underlie this effect. And the first one is that the fluorophore itself experience the intensity of the fluorophore itself will change with the temperature. So there's a dependency there. But importantly, the chemical environment that the fluorophore observes is affects the fluorescence intensity. And as you can imagine, a binding event will change the chemical environment by proximity of the ligand to the fluorophore, but also by conformational changes that the fluorophore, that the ligand may introduce to the protein. And so all of these together mean that as a binding event happens, the fluorescence will change, and that's what we can measure using microscale thermophoresis. Another technique that uses temperature to derive binding data is thermal shift. And there are several variations of thermal shift out there. The most commonly used one is differential scanning fluorometry. And here we look at the transition of the protein in the presence of a fluorescent dye. So typically a natively folded protein will have most of its hydrophobic residues in its core, and a fluorescent dye will be freely available, giving rise to a very low signal. But once the protein starts to unfold, the hydrophobic residues become exposed, the fluorescent dye can bind, and that increases the signal until the protein is completely denatured. And from this, we can derive a melting temperature and obtain data on the thermal stability of the protein. There is another way of measuring this called nano-DSF, and here we would be looking at the intrinsic tryptophan fluorescence rather than using fluorescent dye. So the thermodynamic equilibrium changes of a protein changes once you add a ligand, and this means they can become either thermally more stable or less stable, and that's what we look at when we look at binding events. This technique is more low to medium throughput depending on the instrument available to you, but can measure up to 4,000 samples per day. However, you will be unable to obtain any binding affinity data. The probably most commonly used, most popular technique is surface plasmon resonance. And that's a technique that doesn't rely on a fluorophore or heat to measure binding. However, it's also not a solution-based technique. Here, the target will be immobilized on the censorship surface before the analyte will be flown over the surface in a flow cell. And the censorship sits on a prism and is hit with this source light, and the instrument and the light is reflected, and the instrument will measure the reflective index of the sample. The binding of the analyte will change this reflective index, and that's what measured in the instrument. This technique allows for real-time measurements of biomolecules, biomolecular interactions, and as there's flow, we can derive several constants from the sensorgram. For example, the association constant as the analyte is blown over over the bound target or the dissociation constant as the analyte is washed off. And the equilibrium dissociation constant, which measures the binding affinity, and also the Amax, which gives you the response where the analyte is saturating the ligand. So this is a very useful technique, especially if you're interested in on or off rates that may be important for a drug discovery program. But in SPR, the signal strength is increasing with the size of the analyte, which is very good if you're looking at protein-protein interactions, but it's less sensitive if you may be looking at fragment interactions. So this is where a company called Cryoptics have brought out the WAVE system, and that uses grating coupled interferometry. And it relies on the same principle as SPR, where a target is immobilized on a sensor chip. But here the light travels across the entire chip surface rather than just a single point. And that means it's much more sensitive. Many more binding events will contribute to the signal, which makes it much more suitable to measure smaller analytes. Another oldie but goodie is nuclear magnetic resonance. Here, what we're talking about is a ligand observed technique rather than a target observed technique. So what we're looking at is the NMR spectra of our ligand and how that changes if a protein target is bound. And typically there's three methods out there that can be used: STD, NMR, CPMG, and Waterloxy. In our lab, we primarily use STD NMR, and in this graph, you've got an example of what this may look like. So in the purple, we have the 1D NMR spectra of the ligand shown above. Then we have to run a control experiment, which contains just the ligand, and we want to make sure that the saturation pulse that we'll be using doesn't already hit the ligand. And once we add the protein of interest, if they bind, then the saturation that the protein experiences will be transferred onto the ligand, and this is what we can observe here. Also useful with this technique is that one can run competition experiments. So if you have two ligands that you think bind in the same site, then if you add them both, in this example, you see a reduction in the peak intensity compared to just having the one ligand, and that suggests that the second ligand is competing with the first ligand for the binding site onto the protein. This technique is also very useful as you can combine several ligands in one experiment, as long as their 1D spectra significantly different enough that you can identify which one gives rise to the signal that you may observe and has a very short assay development time. Finally, still the gold standard biophysical technique, ITC. It's very sensitive, fully label-free, and you're able to measure freely in solution. However, it's still very underused in drug discovery, and that's due to a low throughput and quite large protein requirements. ITC allows for not only determination of binding affinity, but also stoichiometry, entropy, and enthalpy. And when binding occurs, then heat is either released or absorbed, and this is measured in the calorimeter. Usually you have a gradual titration of the ligand into the sample cell that contains the target, and each addition of ligand will give rise, in this case, to a heat release, which is measured. As you add more and more ligand, the target gets saturated until it's completely saturated. Each of these peaks can be integrated and plotted against the molar ratio of the ligand versus the protein, and then you can calculate the binding affinity and various other parameters that way. So just to summarize, then the techniques we've talked about today on this slide, positives and negatives can be found for each technique. As we go from left to right, MST and SPR, and to some extent thermal shift, are more suitable for screening than potentially NMR and SPR due to higher use of protein and throughput. However, SPR, for example, is the only technique that will give you K on and K off rates. So really, there's no one best method out there. We will just evaluate the target and the data that are required. Are we looking at the screen? Are we looking at an orthogonal method with potentially fewer compounds? And what data do we need? And for that, we will pick the right technique that we need to get these data. So in the next part of the presentation, we wanted to give you two examples. One will be microscale thermophoresis, and the other one will be GCI, where we explain how we set up an assay and then how we use that to start off a drug discovery program. In the lab, we like microscale thermophoresis because it's very fast. It's got very dynamic, very high dynamic range, which means while we might have a fragment screening project where we probably expect to get high micromolar, millimolar affinities, we might have a control ligand which has a low nanomolar, picomolar affinity, and these instruments will give us the flexibility to measure both in the same run. We don't require much protein, and importantly, we can also look at challenging, potentially tertiary systems, for example. As Kat already mentioned, we have got both a monolith system, which uses glass capillaries, and a dianthus system, which uses a microtiter-based, which uses microtiter plates. So when we look at developing a new assay, we'll always have to start off with target labeling, of course. Then we'll look at binding affinity determination of our positive controls, assay optimization, and stability evaluation, of course. So when we talk about target labeling, we've got two options to do so. The first one would be using covalent linking via cysteine or lysine residues, and the other option would be to use affinity labeling via his tags or SNAP tags. And then we look at the labeling efficiency. We always aim to be somewhere between 0.5 and 1 dimolecule per target protein. And then we'll check the behavior of the protein to ensure that it still has the behavior that we're looking for. One thing that can happen, as you can imagine, is that the protein aggregates. And the nice thing about MST is that we can see that in the trace straight away if we observe these bumps compared to a nice smooth diagram of a well-behaved protein. That means we have some aggregation. And usually the quickest way to improve this is by changing the labeling strategy. The other thing that we look out for when we use the monolith capillary-based system is absorption to the glass capillaries. What happens is that you will get like a bat shape rather than a sigmoidal curve, meaning that lots of your labeled target proteins will stick to the glass capillaries, and that will prevent you from determining any binding affinity. So one way of changing this is by using especially premium coated capillaries, or again, by just changing the labeling strategy. And finally, we will look at checking that our protein actually still binds the positive control ligand as we would expect. So we'll just run our target protein in the absence and presence of ligand and look that we get a nice difference between these two. And as I said, if this isn't the case, again, in the first instance, the quickest way of improving this is by changing the labeling strategy. The next thing we do once we're satisfied that we don't see any artifacts and we have observed initial binding is determine the actual binding affinity of our positive control ligand to check it's in the expected range. And what we hope to see is a nice distribution of the traces across the titration series and a nice curve. And if this is not the case, then we would look at things like incubating the sample, checking the buffer conditions, and possibly, again, adapting the labeling strategy. But once we have observed binding in the expected range, we still always try to optimize the assay to the maximum for various reasons. One is that we want to get the maximum signal window with a stable assay to be sure to have a very robust screening assay, but also an optimized buffer will help to prevent and reduce aggregation of the protein, which might be becoming more important during a screening campaign when you don't just spend 5 minutes measuring a sample, but it might take hours to complete a screening campaign. And when we talk about buffer optimization, we mean looking at different buffers, looking at salt, so ionic strength, it could be different detergents, and it could be additives such as calcium or magnesium that can make a huge difference. And at the end, we will look at target stability. So as I already mentioned, a screening campaign may last a couple of hours or take a couple of days. So we want to simulate that our positive control will give us the same signal window and binding affinity across the time it would take for us to do this assay. So we'll run the same sample with over a time course, and we'll hope to see stability there. And if this is not the case, then we may go back to buffer optimization, or we might have to think about how we adapt our screening mode in order to ensure that we still obtain robust data. And with this, I'd like to take you through one example of a fragment screening project where we've used microscale thermophoresis. And this is a project we've worked on using lysine methyltransferases, and these are epigenetic targets involved in gene regulation, and they covalently modify histones. So they will put a methyl group onto the lysine residues on histone proteins. And in the lab, we've produced a functional catalytic domain of a KMT, and we've been able to show via thermal shift that it still binds as cofactor, that it binds a cofactor SAM. However, we couldn't use thermal shift as our primary screening method, as this is a tertiary system, meaning it doesn't only bind its cofactor SAM, but it also obviously binds the histone protein. But once you've bound SAM, you won't observe any further stabilization of the protein to pick up any ligand that would bind in the peptide site. And we were particularly interested in this peptide binding site that we wanted to identify any new hit matter for. To help us along the way, we've also had a crystal structure of this protein available. So we've used as a primary screening assay microscale thermophoresis, And we screened 320 fragments in this assay, obtaining 15 hits with good binding affinity, ranging from 19 micromolar to 1 millimolar. And then as an orthogonal method, here we've used STD-NMR. And the hits that came through the STD-NMR, we've then taken into crystallography to confirm that they indeed bind into the peptide site as we'd anticipated. And then we've taken them through into analog synthesis to improve the affinity of these initial fragments and drive the drug discovery program. So this is an example of what a primary screen might look like, performed via microscale thermophoresis. So what we do is we always screen DMSO reference and our positive control. And this was done using the capillary-based system. So we'll set a hit threshold. In this case, it was a minimum of 3 standard deviations from the mean of the reference, and that's depicted in this pink area here. And all the grayed out samples are compounds that are classified as non-binders. The yellow ones are our positive controls, and all the light blue ones are our hit matter. And as you can see, they can be above or below our non-binder area, and it doesn't matter which way the signal goes, and it also doesn't matter how far away from the inactive area these are. So distance does not correlate to affinity in this technique. What is also particularly nice with MST is that we get an immediate false positive detection. For example, we can pick out proteins that cause ligands that cause aggregation. We're seeing these bumps in the curves again. We can pick out compounds that, for example, induce photobleaching by seeing a decrease in the baseline signal, or we can pick out compounds that may cause autofluorescence. And as I said, we had 15 hits identified, and 7 of these we've taken into STD-NMR, and we could observe binding for 5 of these. And they then went into crystallography, and we identified 3 of these fragments bound into the peptide channel as we had hoped. And using this, we could identify attachment vectors for growth down the peptide channel. And in this case, we used docking to identify molecules that we would then synthesize with using focused arrays. And in one round, synthesizing 20 molecules using this focused arrays, we achieved a 10-fold reduction or a 10-fold increase in affinity. And this is how we started off this hit identification program and moved it forward into hit expansion. And I think this is all we wanted to say today about microscale thermophoresis. And now I'll hand over to Alec to tell you more about graded coupling interferometry.
Good afternoon. In this, that's not me. Right, I think I've got control. In this section, I'll discuss the principles of grating coupled interferometry, or GCI for short, and how to set up a successful experiment to measure binding affinities using GCI. As previously mentioned, GCI is similar to SPR in the way that you attach a target to a chip and flow analytes over the top in order to measure binding events. The machine that we use for GCI is the Wave Delta from Creoptix. And the main difference between the 2 techniques is the direction of the evanescent wave being horizontal in the GCI, but being at the angle of total internal reflection for SPR. The theory of GCI can be quite complicated, but in brief, a laser is diffracted into a waveguide via a grating, creating an evanescent wave. A waveguide is a structure that restricts the transmission of energy to one direction. This evanescent wave then travels the length of the chip over the top of the immobilized target. Any binding events of your analyte to your immobilized target are seen as a change in refractive index and are then translated using interferometry against the laser's reference beam. And then this interference pattern is then translated in a time-resolved intensity signal that is a facsimile for association and dissociation.
Alec, I think I have control here, so I shall just click to the next slide whenever you ask.
Okay, if you click. In SPR and traditional kinetics, usually you'd run a dose response curve of your binding partners across the surface and detect binding at different concentrations. However, the wave delta can use Creoptic's proprietary rapid mode, which instead of injecting multiple concentrations, injects one concentration in pulses of different times. And this can give exactly the same data as the traditional kinetics of Ka, Kd, on and off rates, but it does so in a fraction of the time. So it's much easier to run multiple samples in a day in comparison to the traditional method. And on top of that, it involves much of the compound handling that you have to do beforehand, creating your own dilution curves, as the machine essentially does it for you in the machine. So move to the next slide. I'll now discuss the plan and setup of a GCI experiment. So if we move again. The first part of any GCI experiment is the immobilization of the target to the surface of the sensor chip. There are various sensor chips that you can purchase from Creoptics for use in this machine, but there are two main methods of immobilization: direct coupling, which is using the amino acid side chains to directly couple to the chip surface, or indirect coupling, which is via an affinity tag on the target, such as a His tag. If we move to the next slide. Thanks. The two methods both have pros and cons. Direct coupling gives a stronger, more consistent attachment to the chip, but it's also a random attachment, as the target can be bound through any of the amino acid side chains that you've chosen to link, so any amine bond on lysines. And this obviously, should there be a lysine in your active site, could mean that you've blocked the active site through steric hindrance. To avoid blocking the active site, as in the examples, you can use indirect coupling, and this will couple the protein in one orientation using only the His tag. The issue with the indirect coupling is that your target may be lost over time as the binding isn't quite as strong. Next slide, please. So once you've chosen your immobilization method, there's a number of factors that need to be optimized. Firstly, the immobilization level needs to be considered. And by this, I mean the number of target molecules actually deposited on the surface. The immobilization level can vary depending on the purpose of the experiment. So fragment screening requires a higher mobilization level due to the small size of the fragments and the need for many fragments to be bound in order to get a strong signal, whereas protein-protein interactions require very low mobilization levels in order to get the best data out of them. For direct coupling, the pH of the buffer is also very important, as you need to get a net positive charge on your target to be immobilized. And one way to do this is to use a low pH buffer. And the idea of the pH scouting is in order to get a quick, strong attachment at a low pH, but not a pH that's so low that it denatures your protein before you've even managed to attach it to the chip. For the running of binding partners, the matching of running buffer and sample buffer is essential to avoid spikes in the trace and solution-based effects. Another important factor is selecting the correct blank. This could be an activated and then deactivated channel, or it could be an unrelated protein that you are sure that your binding partners won't bind to. Next slide, please. So once the experiment's complete, the software produces traces such as you see in the top left. From these, you can calculate the software automatically calculates the Ka or on rate, the Kd or off rate, and the big K little d, which is the dissociation constant. And this is the data that you would report forward to other areas of the project and is the main takeaway from GCI. At this point, you can also assess other things such as experimental artifacts. So sometimes you get spikes in the data that can be precipitation or aggregation. And there's things such as mass transport effects, which occur when the binding is limited by the diffusion of the analyte rather than the on rate of the binding reaction. Next slide, please. So the wave itself has a sensor chip that has 4 channels, and this can allow you to screen up to 3 targets in parallel using the same initial starting binding solution. And then the 4th channel would obviously use as a blank. So this can be very useful. The wave itself has 2 plate holders, so you can screen 384 wells in one shot, and each well will be passed over all 4 channels in parallel. So next slide. I'll now briefly show an example of a project where we've used the GCI and other techniques to move the project forward and find some good hits. So in this project, we began by screening 1,100 fragments through MST. From this, 106 hits were selected, and these were taken forward to be counter-screened in GCI and using the NanoDSF. So next slide, please. So here we've got some example data from all 3 biophysical techniques. The fragment at the top shows a good MST curve, and the KD calculated from this is very similar to the KD calculated from the GCI. And as you can see, we've used the rapid mode in order to save time and compound costs. For this top compound, you can also see in the NanoDSF, there's a strong shift in melting temperature. And essentially this hit has come up as a hit in all 3 techniques and is a strong hit. The second fragment, however, shows a fairly good KD in the MST and a matching responsive KD in the GCI. However, the thermal shift detected by a NanoDSF would be considered insignificant as it's not far enough away from the DMSO blank. So if we move to the next slide. So as you can see, fragments may present as hits in a number of complementary techniques, but not all hits will detect all fragments. So it's very important to run orthogonal techniques to get orthogonal hit confirmation between the techniques. So the Venn diagram shown here basically is the summary from this experiment, and only 26 hits out of the 106 were seen in all 3 techniques. So had you only chosen to do the NanoDSF or the GCI alone, or even the MST alone, there's a lot of hit matter that may have been missed. The difference usually can be explained between the difference in the techniques. So things like MST are in solution, whereas GCI is an immobilized protein. So it's important to see where the differences between techniques can work together in order to give you a full picture of hit matter from fragment screens. And that's me done.
Thanks, Alec. So just to add a couple of notes, just to conclude here. Just a few concluding remarks. So hopefully the descriptions today have lifted the lid a little on the various biophysical screening techniques and provided some useful examples of how each can be applied with the different benefits of each, and show how this fits with the drug discovery process. and what considerations there are as you go through as to which method to choose. Of course, you know, we are here at DomainX to answer any queries you have, not just within this webinar, but at any time, please feel free to reach out and contact us if we can help you with anything, biophysics and beyond. Just to say, you know, there is more to it than the technology. You know, we are very proud of the people that we have here at DomainX that drive these technologies in biophysics and also in our other disciplines. And the relationships that we create is by putting our heads together that we get the most out of these technologies and work out which one is the best fit for a particular project. And also, you know, the shared mindset and philosophy of wanting to find solutions and work collaboratively to apply these techniques to best effect. So just briefly, if you wanted to find out any more about what we do and work out how to contact us, then it's all there online. It's all there on our website, along with some further case studies and examples and white papers. And if there's anything that's really grabbed you there, and you think, you know, actually we want to be part of that, then we are growing and you know, we are recruiting. And more can be found about the positions where we're recruiting on our website as well. And that's including into our assay biology and biophysics team. So thank you very much for tuning into our webinar today, and thanks, Kat, for hosting us. And back to Trevor.
Thank you very much, Kat, Jana and Alec. I'm sorry about the audio problems on Kat's end. I hope people managed to catch most of what she had to say today. Just looking at the Q&A, we haven't had any questions posed by the audience, so obviously the team did an excellent job in explaining very clearly what they did. I actually have a question which I'd like to ask. I know that you've stressed the fact that often it's useful to use a number of complementary techniques, but you've got to start somewhere. So how do you decide which biophysical technique might be the most useful for any given protein target?
Shall I go? I think we'll look at the target first of all, foremost. Look at, you know, what kind of target are we dealing with? Are we dealing with the membrane protein? Are we dealing with a protein that might have lysine residue in the active site, so it will exclude us to do a certain covalent labeling method, and then we actually tend to test more than one technique at the same time in parallel, and the one that, you know, gives us the best data we'll probably choose as the screening method of choice if we have flexibility on that.
Okay, and I have a question for Alec. You talked about GCI, which of course is going to be a pretty new technique to a lot of people who may be familiar with SPR. I think you implied, but maybe important to spell out, that it's got the same flexibility as SPR in order to determine K on, K off, and KD in a single experiment. But just doing a bit more compare and contrast between GCI and SPR for us, you know, what do you think are the strengths and limitations of the two techniques? And you're aware of situations where GCI has worked where SPR has not, or vice versa?
So in terms of technique and output, it's almost identical. The traces look the same, and the on and off rates and KDs can all be calculated in the same way. The difference with the GCI, especially with the wave that we have, is because of it being a parallel beam of light, it means it is more sensitive, and you can then get more definition on your fragment hits with being closer to the correct on and off rates, because with fragments, they'll certainly be extremely fast on and off rates. I've done some fragment screening with SPR and the Biocore machines, and they will still give values, but they often then also give an error message saying this is an estimated value and it's very close to the limits of the machine. And this may cause more variation in repeats, whereas the GCI we expect to be more accurate as it's able to work at a much faster rate. And a lot of the increasing sensitivity and increased sensing is also due to the microfluidics in it. So the GCI has microfluidics on the chip, so that the actual valves that let the liquid flow back and forth are like nanometers away from the detection area. Whereas in the Biocore machines, the microfluidics is in the machine and that's stamped down onto the chip, so that they're actually quite far away in terms of distance. So the GCI has the same modes as the SPR does, so you can still run the traditional kinetics with varying concentrations. The main benefit we've found so far of the Wave is this rapid model. Truthfully, it really does cut the time spent working, machine time down by 10%, preparation time down by 90%, and you're able to use at least 1/10 of the actual reagents in order to get the same results out.
Okay, good. Well, thank you very much for that. We still haven't elicited any questions from the audience, so I think it's probably time to wrap up. So thank